right, so we're on to chapter 4 today. Some of you have lab later today. In lab, we're going to be doing cell structure, so we'll go along with what we just did in chapter 3. Okay, so chapter 4 is a whole chapter basically on understanding membranes and what they do for our cells. So that may seem crazy, but it turns out membranes are really, really important. I've already mentioned that some of the functions of membranes. So for example, every cell in your body has a plasma membrane. So that's a selectively permeable membrane. So that's like certain things cross and other things are not allowed to cross. Your cells actually spend a lot of energy transporting things across the membrane. So we're going to sort of dive a lot deeper in this chapter into talking about these things. So again, the plasma membrane is this membrane that separates the internal environment of the cell from its external environment. And this is necessary to maintain homeostasis. It regulates the transport of molecules in and out of the cell. So we've talked about this already, but when you think membrane, you should think phospholipid bilayer. And I'll draw a picture of what that looks like. We've already talked about it a bit, but in most of the diagrams you'll see we'll have the inside of the cell as being downwards in the diagram, and the outside is usually upwards. So this is the intracellular solution. Inside of the cell, there's the cytoplasm, and that consists of the cytosol as well as the organelles. And then the outside, of course, is the extracellular space. And that is also referred to as the interstitial fluid. Interstitial fluid. And that's a very important fluid because it bathes our cells. It has to contain oxygen and glucose and other nutrient molecules, as well as things like amino acids, which are used to build proteins. It may contain fatty acids, can be used for metabolism and so forth within the cell. So this interstitial fluid is very important for the health of our cells. All right, what does this lipid bilayer look like? Well, remember what a phospholipid looks like. So our sort of cartoon version of a phospholipid is it has a polar region, which is referred to as the head, and then it has a non-polar region, which is referred to as the tail. The non-polar region consists of 2 hydrocarbon tails. And the polar region is always it's gonna, remember there's water inside and outside, inside and outside of your cells. So the polar regions of these molecules will always orient themselves towards the water. And the non-polar region of the molecules doesn't like water, they're hydrophobic. So they're gonna be oriented away from water. And so this is a bilayer because it's 2 layers, right? There's a layer like this facing inwards, and a layer in which the polar ends of these molecules faces outwards. So the main point here is we have this huge region in the middle, which is hydrophobic. So remember that means water, hydro means water, and then phobic means fearing, right? So there's this water-fearing region. So it's difficult for water can pass the membrane, but it's relatively difficult. So we'll talk about today about how water can go through your plasma membrane, even though we have this large hydrophobic region in the middle. Ions have a very hard time. Remember ions, let's do sodium. It has a charge of +1. So sodium generally, if you have water, the water is going to be oriented this way. Can anyone tell me why? I mean, obviously the water molecules are bouncing around, so it's the sodium. But if you're somehow able to take magically a snapshot, this is supposed to be oxygen, by the way, right? That's the O. The oxygen end of H2O is often pointed towards the sodium ion. Why would that be?
Oxygen is negatively charged.
Yeah, exactly. So our oxygen has a partial negative charge. Do you remember we used the Greek letter delta to indicate that? So we used the little delta, and we indicate it's negative. And the hydrogens, again, we can use the Greek letter delta or positive. So because of that, again, it's kind of like if you played with magnets when you were a kid, this north and south poles like to snap together. This is a little bit like that. Of course, it's different in the sense it's charges, right? This has a charge of +1. These are partial negative charges, so they tend to want to orient it towards the sodium. And if we did chloride, which has a negative charge, I think you can guess where we're going with this. It's going to be just the orientation of the cell of the oxygen. I'm sorry, the water molecules will be different, right? It will be with the hydrogen, which is a partial negative charge. Sorry, partial positive charge, excuse me. Oxygen has the negative charge. Okay, my negatives and positive. Partial negative charge. Hydrogen has a partial positive charge. It's going to want to associate a little bit with the chloride. Now, of course, they're all moving around, so it's not like they're static, right? But so this means that ions really like water. They're hydrophilic, right? And water loves to associate with ions. But this means, conversely, that ions have a very hard time with hydrophobic regions. So it's very difficult for polar molecules like water to go through this hydrophobic region of a lipid bilayer. It's very difficult for ions to go through this hydrophobic region of a lipid bilayer. So they're going to turns out their cell will have a number of tricks for getting things across the membrane. So we'll talk about that. Okay, so let's go back to the slide here. We have the phospholipid bilayer with embedded proteins. There's hydrophilic, water-loving polar heads. They face inside, right, and outside. Where there's water present, hydrophobic, water-fearing, hydrophilic, non-polar tails. They face each other in the interior of the membrane away from water. Now, there's also steroid. Cholesterol is a steroid, and it's found in animal cells, and it stiffens and strengthens the membrane. But let's draw cholesterol in here. So cholesterol will be a molecule that's going to be you'll see beautiful pictures. I'm just doing a squiggly line, right? But you'll see a better picture in a second when we look at the artist's rendition. So you'll see cholesterol will sort of be stuck in between these phospholipid molecules. And they're oriented in a certain way. They actually have one region that's actually polar, and that sort of tends to stick out towards water a little bit. The rest of it's entirely non-polar, so it's sort of deep within this hydrophobic core of the membrane. We'll see pictures of that. So it turns out cholesterol is very important. So it's so important that your cells can even synthesize cholesterol if need be. It's important for all your cell membranes to maintain the proper fluidity of their membranes. We're going to talk more about that in a second. So here's what your plasma membrane looks like. So far, I just drew the phospholipid bilayer, but if you look at this picture here, you can see a lot of other things. So we have certain proteins that are called integral proteins. So why are they called integral? It's because they're integrated within the plasma membrane itself, right? They sort of sit within the membrane. There are other ones that are called peripheral proteins, and that's because they're on the periphery, right? Typically, they're going to be on the cytoplasmic side, although it's possible you might have one on the outside, but usually they're on this side. They often associate with another protein, so they might have some affinity, meaning they're likely to bind in a non-covalent way, using maybe hydrogen bonds or ionic bonds with, say, this protein here, and they're going to be sitting there. So we have integral proteins, peripheral proteins. Now we have some of these proteins that stick out into the extracellular space, which is shown up here. And so we have proteins, and they have actually, they have carbohydrates attached to them, and for that name, they're called glycoproteins. So protein, of course, comes from the fact that part of the molecule is protein sitting within the membrane, and the glyco, that term means carbohydrate. Okay, so you can see that this is a chain of essentially, you see these are monosaccharides, things like glucose, things like that, stuck together. and they're sticking out from your cells. And so that's what your cells look like. They have all these little carbohydrates sticking out. I think we might have talked about these before a little bit, but what might be a good guess for why we have all these carbohydrates sticking off your cells?
Oh, wait. Identification.
Identification. Yep. Go ahead. Keep continuing what you're going to say. Go ahead.
Transfer in between the cells, but that's protein.
No, we're gonna talk. So this is actually for transport. We'll talk about that. Like, but you're right on identification, right? So these things that stick out. So carbohydrates can make. Remember, there's different carbohydrates, right? There's different monosaccharides. So you can make these really unique structures that are branched and so forth. And they're kind of like a little ID, like a little ID tag sticking out of your cells. So they tell your immune system, hey, this is me. It's like a little flag. Don't attack me. Don't kill me. Now, bacteria will have other things sticking up off their surface. You've heard of viruses, right? Having spike proteins sticking off the surface, right? So those are things your immune system are going to attack, right? It's like, okay, this is foreign, we're going to attack this. But these are little things to say, essentially tell your immune system, don't attack me, don't kill me, please. And also, if you have two cells that are neighboring each other, they need to know who their neighbors are. So they can actually detect, somehow they're able to use this. I'm not quite sure how they do it. It's sort of magic to me. But they're able to identify each other through these sort of surface interactions with these surface molecules. Now so those are glycoproteins. Okay, here's one that's interesting. Here's a glycolipid. So let's see if we can take that apart. What is it? What kind of molecule is a glycolipid? It's made of two types of molecules we've talked about. Carbohydrate, right? Yeah, oh, yeah, you're going to raise your hand, and then lipid, right? That's probably what you're going to say. Sorry. So lipid and carbohydrates. So these are directly attached to the phospholipids. Is basically what we're saying there. Yeah. All right. So that's all. Oh, since we got this on the picture here, we've got cytoskeletal elements. So cyto means cell. Skeleton, you know what a skeleton is. So it's the skeleton within a cell. Okay. So there are some proteins that are involved. Now, oh, here's cholesterol. Again, this very important molecule. You see how it's got this ring structure? So that's all hydrocarbon. And at one end, there's a hydroxyl group, OH, if I remember correctly. And that's for that reason, it always orients. So that's polar. So that tends to orient towards water, either outside or inside the cell. All right. Let's take a look. There's a video here. I'll take a look at it.
The plasma membrane is the outer boundary of an animal cell. It's made up of lipids, proteins, and carbohydrates. Phospholipids are the major components of the plasma membrane. These molecules have hydrophilic, water-loving heads, and hydrophobic, water-fearing tails.
So I just wanted to stop this for a second. So this is a really good illustration, right, of what a phospholipid looks like. Remember, you have got glycerol here. It's a 3-carbon organic molecule. And the triglyceride is a little bit different, right? You have 3 fatty acid chains, right? Here you just have 2, but this one, this third carbon has a phosphate group attached, PO4, and you see that has a negative charge. And there's usually another. You see, there's an R up here. There's yet another group that's often polar that's attached up there too. So there's some variety because this R group can be different depending on the phospholipids, but this basic structure is the same. 3-carbon glycerol molecule. Glyceride is another word for glycerol. They're essentially identical. Here's two fatty acid chains. Remember, there's a carboxyl group, COOH. There's dehydration synthesis to make this. Then this is just hydrocarbon. It's just nothing with carbon and hydrogen. It's very hydrophobic. And that's polar, so therefore it's hydrophilic.
The phospholipids form a double layer called the lipid bilayer at the surface of the cell. The hydrophilic heads on one side interact with the extracellular fluid, and on the other side interact with the cytoplasm inside the cell. Interspersed in the phospholipid bilayer are proteins. Some proteins are embedded in the membrane and span the bilayer. Others are located on one side or the other, often attached to membrane-spanning proteins. Molecules of cholesterol are also scattered throughout the bilayer. Carbohydrates attach only to the outer surface of the plasma membrane. Most of the carbohydrates are attached to proteins, forming glycoproteins. Glycoproteins often function in cell recognition. The structure of the plasma membrane is often described as a fluid mosaic. The proteins are interspersed among the lipid molecules like tiles of different colors within a mosaic. Many of the proteins are able to move sideways through the bilayer to some degree, giving the membrane its fluid quality.
So that's a good video. So the fluid nature would have been hard. It's nice to see that here. So they mentioned that some of the, so that if it's a fluid mosaic model, the idea is that I think I have a picture actually of some mosaic. Where is it? Well, maybe I didn't put it in there. You all know what a mosaic is. It's basically tiles stuck in like a cement. They had them in ancient Rome and so forth. So the idea is that if you look at this picture, that this is sort of like the cement, the lipid, and that stuff can, the lipids can move laterally. They're mostly not tied down to any location. But the proteins also can sort of move around. Like these would be like the tiles, they can move around laterally. The lipids actually can occasionally, if they have an energy, they actually flip from one side to the other. That happens occasionally also. But they're mostly moving laterally. And now, what happens is some of the proteins you don't want moving. So you'll actually have some proteins attached to cytoskeletal elements, which is not really shown here, but you can sort of imagine these cytoskeletal elements attaching to a protein, and then that protein is locked in place. But a lot of the proteins, as the video said, do move. All right, so yeah, so here's the idea. We have the fluid mosaic model. We have this pattern of phospholipids, steroids, and proteins in the membrane. Both the phospholipids and protein can have attached carbohydrate chains. We just talked about that, right? Glycolipids are lipids with carbohydrates. Glycoproteins are proteins with attached carbohydrates. You have membrane proteins throughout the membrane. Some are peripheral proteins, are only associated with one side of the membrane. And then you have integral proteins that span the membrane, and they can protrude from either one or both sides. So in other words, the integral ones are embedded within the membrane, and these are often able to move laterally if they're not tied down to the cytoskeletal element. Okay, so what kind of membranes, which kind of proteins do we have in the membrane? One very important class of membrane, of protein within our membrane are called channel proteins. So we need these to stay alive. These allow us to move certain substances across the membrane. They may be gated, meaning that the protein can open and close. So what does a channel protein look like? Well, let's draw a picture. So if we look at a channel protein from the side, it's going to look like this. Color. So you're going to have something like this. And then you're going to continue with the phospholipid bilayer on the other side. Okay, so what does this allow? Well, this allows for certain substances that wouldn't have been able to cross through the membrane now to pass through. A really good example of this is the aquaporin channel. All right, so aqua, what does aqua mean? Water, right? Pour it, think of pour. So this is a pore or a hole through the membrane. This allows water molecules. So you get a little H2O and it's able to pass the membrane. Now, of course, it can go either way, right? It's not, there's no, but sometimes there are differences in the concentration of water on one side of the other membrane, and then the water can pass through. So our cells are naturally not all that permeable to water because water is polar. But once you add one of these channels, suddenly water can pass through. And the rate is tremendous, like millions of molecules per second. It's really tremendous. Now, if you were to look down on the membrane, what would this look like? Now, it depends on what kind of breakfast food you prefer. I call it a bagel. But they look roughly like a bagel. If you prefer donut, that's fine. But it's a protein with a hole in the middle, right? So that's what a channel looks like. Now, we mentioned that these proteins sometimes have gates. So the way that works is sometimes you have proteins that proteins are actually able to move. and change shape. We talked a little bit about why that would be, because a lot of the protein structure, remember when we talked about primary, secondary, tertiary, and even quaternary structure? Secondary and tertiary structure is often determined by weak bonds like hydrogen bonds and things like that. And so because of that, these things are actually able to morph and change. And so this channel can potentially open and close. And that would then be called a gated channel. Okay. For now, I'll return this back to being an open channel for water to pass through. Aquaporin channels, as far as I know, are not gated. But there are other types of channels for ions, such as sodium, potassium chloride, that are gated. Okay. All right, so that's a channel. They're very important for allowing polar molecules to pass through the membrane, which wouldn't be able to pass through otherwise, and for ions to pass through. Okay. Carrier proteins. Okay, so what are carrier proteins? These allow the passage of a solute by combining with it and helping it to move across the membrane. So let's do that one next. So I have very limited space, so I have to keep basically drawing over my same drawing. So let's draw a gate, carrier protein. Now these are also called transport proteins. So either way, a carrier or a transport protein, it looks something like this, right? So here's the protein. Now let's imagine we have a relatively large molecule like glucose. Our cells need glucose, right, for energy. We've talked about that before. So here comes the big glucose molecule. It's a 6-carbon sugar. It goes in there. So there'll be a certain binding affinity for the glucose for this side of this carrier protein. Now, it turns out for glucose, there's a lot of glucose on the outside of the cells compared to inside. Can anyone guess why there's typically more glucose outside than inside? Go ahead. What are our cells doing with glucose? What are your cells doing with glucose all the time? Yeah, burning it, right? It's cellular respiration to make ATP, right? That's what you're thinking, right? So in general, there's less glucose inside than outside. There are exceptions. Remember, liver cells can take glycogen and break it down to glucose. So suddenly they'll have a lot of glucose inside, more glucose inside than outside. Okay. But in general, it's the other way. So there's more glucose outside than inside. So this means by just this process called diffusion, there's a chance that glucose is generally you're going to move from an area of high concentration of a substance to an area of lower concentration of a substance. That's called diffusion. We're going to have a whole slide on that and talk about it more, but that's the general trend. So glucose kind of, in quotes, wants to go into the cell if it could, right? But it can't because it's polar. Remember, things like carbohydrates have hydroxyl groups, those OH groups. Oxygen is more electronegative than hydrogen. Therefore, those are polar groups. So it's very difficult for glucose to get across. So glucose binds here. And then what happens is this molecule does something that's almost like magic. It flips. And I'm not like realistically drawing exactly what this thing looks like, okay? But I'm just trying to give you a little bit of a picture of what it might look like. But it's a protein that's it's not a channel, it's never open, right? Between the inside and outside. But it's able to flip somehow within the membrane, or parts of it flip or move or change. And now the glucose by magic, as if by magic, is now on the inside of the cell. And sure, it has a certain affinity. There's a certain binding affinity between the glucose and the protein, meaning they like each other, they want to bind to each other. But that's not 100%, right? We're not talking about covalent bonds or anything. Probably just some weak hydrogen bonds or something. So then the glucose can be released to the inside of the cell, and then your cell uses it to make ATP, as we just discussed, right? So that's what a carrier protein does. Let's discuss though this liver cell. Your liver, remember, takes glucose and it converts it to this polymer called glycogen, which we talked about. It's the equivalent of starch, but in animal cells. So you have a nice meal. Your liver cells are like, thank you. They take the glucose, convert it into glycogen, this highly branched polymer of glucose. It's in your cells. Now it's between meals, and you need glucose in your blood to supply your brain and the rest of your body with glucose. So glycogen is broken down in glucose. There's more glucose inside than outside. So just by this process called diffusion, there's a natural tendency for glucose to want to leave, in quotes, want to, in quotes, leave the cell. If there's this same carrier protein, guess what it'll do? This carrier protein actually has no idea about directionality. So if a glucose molecule bumps into it over here, it'll flip then and do the exact opposite. It'll flip around and it'll transport the glucose in the other direction. Right, so now the glucose is on the outside of the cell, okay? So that very same glucose transporter can move things both into the cell and out of the cell. And the directionality just depends on the concentration difference. It always moves the glucose from an area of high concentration to an area of low concentration. Go ahead.
How does it, like, how you say it, like flip?
Oh, I mean, somehow when this binds, it changes.
Well, I'm
So-
not like the process of it,
Oh.
like what kind of direction does it do? Like, is it like a V like this and then it goes
Well,
like
yeah,
this?
I don't take this too seriously because I don't know exactly what it looks like. I mean, people are working on that, right? That takes a lot of time and energy to figure out exactly what these molecules look like. So I'm just kind of drawing this as one of, it is something like this. The main point is it's not a channel. It's very different from a channel. It's something that actually has to flip. And we know it's not a channel because there's no gap. There's no, molecules can't just pass through this. Like you can do like little fluorescent dyes or something will not pass through this sort of thing. The other thing is much slower. Channels can literally like transport millions of, 100,000 or million or something molecules per second. This is way slower than that, okay? So there's some real differences we know between carrier proteins and channel proteins. But the molecular stuff, you don't want to work on that. You can spend the rest of your life there are people doing very complex molecular modeling and stuff to understand that with computers and stuff, to understand how the proteins actually change and flip around. All right, cell recognition, we talked about that already, right? These proteins help the body recognize when it's being invaded by pathogens. All right. So we've talked about a lot of these already. We have a channel protein here in A. It allows a particular molecule or ion. So the molecule could be something like water. Ions could be something like sodium, potassium, and chloride. Across the plasma membrane. Cystic fibrosis is interesting. So it's an inherited disease that's caused by a faulty chloride channel. So remember the atom chlorine, it changes its name to chloride when it's in the ionic form, so Cl minus, it has a charge of -1. Basically, these people have a faulty channel. They're not able to move chloride effectively across their membrane. And because of this, they're not, it's a little complicated to get into right now, but because the chloride can't pass across the membrane very easily, water ends up not moving. And so mucus is actually generally quite watery, right? We think of mucus as very thick, but that's because most of the mucus in your respiratory system is actually very watery. It has a little bit of stickiness because of this protein called mucin. But if it's too thick, then all this mucus collects in the airways, and that leads to coughing and all sorts of respiratory problems. So people with cystic fibrosis have a higher incidence of respiratory infections and things like that. Okay, and there's actually we have mucus in other places, like in the pancreatic and liver ducts. So those tend to get blocked if you have very thick, less watery mucus, which happens to people with cystic fibrosis. Okay, all right. What else have we looked at here? We looked at carrier proteins. These selectively interact with specific molecular ions. So I should have mentioned that. So the glucose channel carrier protein is really specific to glucose. It potentially might move another sugar that's very molecularly similar, like fructose, but in general, it's going to move just glucose across. It wouldn't move, say, sucrose, something very different like that. Same thing for these, selectively interacts with a specific molecule or ion, so they can cross the plasma membrane. The family of GLUT. Okay, so GLUT. GLU is for glucose, T is for transporter. Okay, the family of GLUT carriers transfers glucose in and out of the various cell types of the body. Different carriers respond differently to blood levels of glucose. Oh, and then over here, we've got this glycoprotein, the MHC, a major histocompatibility complex. Glycoproteins are different for each person. So organ transplants are difficult to achieve. So this actually has a lot of medical implications, right? Cells with foreign MHC glycoproteins are attacked by white blood cells responsible for immunity. So that's one of the things we have to overcome when you do things like organ transplants. All right. Now there's other proteins that are very important. There's receptor proteins. These are proteins that have a shape that allows specific molecules to bind to them. This binding then causes the receptor to change shape to initiate a cellular response. So let's draw that. So you have, let's do, well, okay, there's different types of receptor. There's a huge variety of receptor proteins, but it's typically going to be some sort of transmembrane protein. It's gonna, and they usually, the book will show it has a little sort of notch in it or something. And what will happen is you'll have a molecule. Now this could be a hormone, it could be a neurotransmitter, but some sort of molecule that will bind, that can bind with a certain affinity to this receptor protein. And this will then initiate changes within the cell. So sometimes, for example, these receptor proteins might be channels. And one possibility is when this molecule binds to the receptor protein, it might open the channel or potentially could close a channel. So there's lots of different things. The other possibility is this receptor protein can be activated. It might have enzymatic activity. It may cause something to happen within the cell. So there's really a lot of huge variety of different receptor proteins that work in different ways. Binding causes the receptor to change shape to initiate a cellular response. Enzymatic proteins carry out metabolic actions directly. So there's this thing called the electron transport chain, which helps with aerobic respiration. And these proteins are located in the inner mitochondrial membrane. Remember, the mitochondria have two membranes, an outer and an inner membrane. Okay, here's a receptor protein. Shaped in such a way that a specific molecule can bind to it. Some types of dwarfism result not because the body does not produce enough growth hormone, but because the plasma membrane growth hormone receptors are faulty and can't interact with growth hormone. So that would then lead to dwarfism. Here's an enzymatic protein. This is you see how it's on the intracellular surface here of the membrane. It's going to catalyze a specific reaction. The membrane protein adenylate cyclase may be involved in ATP metabolism. Cholera bacteria releases a toxin that interferes with the proper function of adenylate cyclase, which eventually leads to severe diarrhea. Okay, so all those things we're talking about, very microscopic things, but it turns out these have big medical consequences. Okay, so let's talk about membrane permeability. So the plasma membrane can regulate the passage of molecules into and out of the cell because it is selectively permeable. Which molecules can freely cross the membrane, which may require carrier proteins, and/or energy depends on the size. So smaller molecules tend to have an easier time getting across the membrane than large molecules. The nature of the molecule, we already talked about that a bit, right? So polar molecules, such as water and glucose, cannot cross the membrane. Charged substances, such as sodium, potassium, chloride ions, cannot go through the membrane without the aid of something like a carrier protein or a channel. Small uncharged molecules freely cross the membrane. So here's good examples of ones that are uncharged. So we have oxygen. Oxygen is nonpolar. We've discussed why that is, right? Because you have two hot oxygen atoms bound together with a double covalent bond. They're both electronegative, but they're equally sharing the electrons. Carbon dioxide, you have unequal sharing of electrons between carbon and oxygen. Oxygen atoms pull on the electrons more, but carbon dioxide, I think we've talked about this before. Carbon dioxide is a linear molecule. So because of that, it's actually considered to be nonpolar. Glycerol actually can get through the membrane, even though So glycerol, we've talked about, this is the 3-carbon organic molecule that's found in fatty acids and phospholipids. It's actually related to things like alcohol. We can talk about the OL at the end here. It actually does have what are they called? Hydroxyl groups, the OH groups, which are polar, but it's because it's so small, it has some ability to get across the membrane. Alcohols also have some ability. There's, because they're small enough molecules, even though they're a little bit polar. Okay, so you can see there's a little bit of a range, right? Depends on the molecule, the size, and how the molecule is polar or charged. So if these are small enough, they can sort of slip through the membrane. And what drives these things across? Well, there's something called a concentration gradient. When you have, let's use the example of oxygen. Typically, our cells use oxygen for aerobic respiration. So there's typically more oxygen outside of a cell than inside. So that means there's a concentration gradient. So oxygen will tend to move down its concentration gradient from outside to inside of a cell. And that's through the process called diffusion. Okay. All right, we've already talked about this. Water is a polar molecule and cannot readily cross the membrane. Therefore, I drew an aquaporin channel on the board. These are special channels that enable water to cross the membrane. So large molecules, ions, and charged molecules are unable to freely cross the membrane, but can cross the membrane via channel proteins, which I discussed, that support pore in the membrane. Think of like a bagel or a donut, whatever your favorite breakfast food is. And then carrier proteins that are specific for a substrate. And again, those, as I showed on the board, those actually change their shape. And then finally, we're going to talk about something for some large molecules, really large molecules like proteins and things. We're going to have to do something quite different. Those are going to be too big for channel proteins. We're going to have to use what are things called vesicles, which are these spherical bits of membrane. And they're going to do processes called endocytosis and exocytosis, which we'll talk about in a second. So here's just the summary diagram of what we've been talking about. We've got charged molecules. You see this, how this arrow bends back? It means they can't go across the membrane. But if you have a specific channel protein for that charged ion, such as sodium, then they would be able to go across. Here's water going through an aquaporin channel. Water's polar, can't get through. Uncharged, non-charged molecules, like a lipid, they can get through quite easily, like maybe a fatty acid. It does have a carboxyl group on one end, which is polar, but in general, they're very non-polar. So a fatty acid might be able to slip through the membrane. A very large macromolecule, like a protein, would not be able to get through. It's just simply too large. Okay. So how are these molecules moving? Well, you've heard about this before, I'm sure, in high school. There's this process called diffusion, which you have a movement of molecules from an area of higher to concentration of that particular molecule to an area of lower concentration of the particular molecule. So we say that the molecule is moving down its concentration gradient. So let's look at the picture here. So we have these two poor goldfish. I don't know why they're goldfish in here. What they have is a sugar cube, and they put that in food color, so purple food coloring. Now, in the very beginning, the dye, and of course there's probably sugar in here too, are interspersed, but they're very packed together, right? And here's the water molecule. Now, what you have to imagine is water molecules are bouncing around all the time because of thermal energy, right? And because of that, they start to break off these dye molecules. And then just by random chance, a given dye molecule may actually move away from this sugar cube where it was, or it may move back, right? They're going to move back and forth. But over time, just by random movement, the dye molecules will spread out, okay? You can think of it this way. What if I had a box, like I had a shallow cardboard box, and I had a bunch of white marbles and black marbles. And I spent a lot of time putting all the white ones on one side and all the black ones on the other side, right? Now I just shake the box, and over time, what would you expect to happen? If you came back a few minutes later, they might be all mixed up, right? Now, if I keep shaking for long enough, are they going to sort themselves back again? So all the white ones on one side, no. Theoretically, that's possible, but it's very highly, highly unlikely, right? Because of the random nature of the movement. I'm probably going to have to put energy in to sort them back out again, right? So this is essentially this process that's going on here. It's diffusion. So in diffusion, essentially things get mixed up, right? In this case, these purple dye molecules get mixed together with the water molecules, and it's quite difficult to go backwards, right? So that's what's happened here. I don't know why they put fish in this bowl. That seems kind of cruel. But you can see the purple molecules are moving down their concentration gradient from an area of high concentration of purple molecules to an area of lower concentration. Eventually you get to the end where they're spread out evenly, right? Now, there's another point here though. What about the water molecules? In the very beginning, the water molecules have a high concentration away from the sugar cube, but they're very low concentration inside the sugar cube. So the water molecules actually move down their concentration gradient also, right? Just like any other molecule, water will move down its concentration gradient from an area of high water concentration to an area of low water concentration. Okay. All right, so each molecule will move down its own particular concentration gradient. And eventually equilibrium is reached. Now, in the very end here, diffusion is still occurring, right? Molecules are still diffusing back and forth. But what when we say diffusion in this course, in the book probably also, what they're really referring to is net diffusion, which is when we have a net movement of something from one area to another, right? All right, so for example, when a piece of crystal dye is placed in water, the water molecules move until equilibrium occurs. Once the solute and solvent are equally distributed, the molecules continue to move about. So there is kind of a diffusion there, but there's no net movement in either direction. So in a sense, we don't have net diffusion anymore, right? So at that point, all right. So why is this important? Well, this is how we breathe. So gases can diffuse through membranes. Oxygen and CO2 enter and exit this way. So what happens is this: water. There's inside your lungs. You have these little teeny sacs called an alveolus, plural alveoli. They have a very thin layer of water. The actual oxygen actually dissolves into that water, and then from there it can actually go through the plasma membrane of the epithelial cells that line the alveolus. And then from there, they go through the next membrane, through a small interstitial space, and then through the membranes of the wall, the cells that make up the wall of the capillary. So it's actually an extremely short distance. So the oxygen can move quite rapidly by diffusion from the air in your lungs into your cardiovascular system, into your capillaries. So what affects things like diffusion? And CO2 will move the other way. So what are some factors that influence the rate of diffusion? There's temperature. The higher the temperature, the higher the rate of diffusion. Pressure, the higher the pressure, that indicates more oxygen, for example, the higher the rate. You can do things like electrical currents, potentially, that can affect these things. Also, molecular size. So small molecules tend to diffuse faster than large molecules. Okay, so let's talk about osmosis. So, well, let me introduce osmosis first before we talk about this. So we actually saw a sort of a preview of osmosis here, where we saw water moving down its concentration gradient from an area of high concentration to an area of low concentration. So with osmosis, the only difference is we're now talking about the movement of water across a selectively permeable membrane of some sort. So osmosis. So let's imagine we have something like this, and you'll see it also in the video, but let's imagine we have a bunch of water molecules here. I'm drawing them really big, but okay, I think you get the idea. So there's a lot of water on this side. Imagine it's just pure water on that side. Imagine on this other side, we have a sucrose solution. So intermixed with the water molecules is a lot of sucrose. We made it very concentrated. So, okay. Well, Maybe put too many of this side. Let's see. I'll pack this one side up a little more. All right, so because of all the sucrose, which side, the left or the right has more water molecules? The water molecules are the blue molecules. My diagram, which side has more water molecules per volume? Yeah, this side has more, right? The way I drew it. This side has less because the sugar molecules, sucrose, is taking up space, right? Now, what I've done here is we imagine we have a membrane here. I didn't really draw this perfectly, but imagine that sucrose is a fairly large molecule. It's a disaccharide. Imagine that it cannot get through this membrane, but the water can. So each molecule just pays attention to its own concentration gradient. There's more water on this side than this side. Now, I'm saying that this membrane, some sort of plastic membrane, is a porous plastic membrane. The water molecules can get through, right? So they can go either direction, right? So just by random chance, some water molecules will go from right to left, but some molecules will go also from left to right in this diagram. And because there's more water molecules on the left, we're going to end up with more movement from left to right than from right to left, just by chance. And so what we're going to do, what we're going to get, that's essentially osmosis. Water is moving down its concentration gradient from an area of high water concentration to an area of low water concentration, okay, across a membrane, selectively permeable membrane. So that is in fact osmosis. So let's take a look at the video.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a semi-permeable membrane is called osmosis. If a molecule such as urea is added to one side of the membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. Because the concentration of water molecules is lower on the right-hand side, there is a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
So they introduced a little bit of an extra idea of this idea of free water molecules, but it's essentially the same thing. Even if you used a non-polar molecule, unlike urea, sucrose is actually a little bit polar because of OH groups on it, hydroxyl groups. But even if you had a molecule that didn't even have any, wasn't polar at all, but it's just simply taking up space, you would still have this phenomenon where you would have less water on this side than on this side, and therefore you get osmosis. Okay. So the basic rule is the same, which is that water will always move down its concentration gradient from an area of high concentration of water molecules to an area of low water concentration of water molecules. So sometimes books will flip this around and say, well, what about this? What happens with the solute concentration? So in this case, I have two solutions here, right on the left and the right. I have no solutes on the solution on the left, right? It's just pure water. On the right, I have the solute. I said the sucrose earlier. Let's stick with sucrose. So there is a higher solute concentration on the right compared to the left. So because of that, that it affects the water concentration. But effectively, what happens is water will always move If you have two solutions side by side and they're separated by the selectively permeable membrane that allows water to pass through it, but not the solute molecules, then water will always move from an area of low solutes concentration to an area of high solute concentration, right? So I have to state that because sometimes books will present it that way also. Okay, so here's a picture here. We have the diffusion of water across the differentially permeable membrane. It occurs from higher to lower concentrations of water and toward a higher solute concentration all across the selectively permeable membrane. Since the solute can't cross the membrane, it draws. So this, I don't really completely agree with this. Like, the solute doesn't actually draw the water, right? Solute can't draw the water. I mean, that's effectively what it seems like, right? It seems like the sucrose here is drawing the water. Water isn't really being drawn. It's just moving down. It's blindly moving down its water concentration gradient, right? So I don't completely agree with that. You'll see that in a lot of textbooks and stuff that use that sort of terminology. It sounds as if the solute like it knows something. It's like pulling the water towards it. It's not really what's happening. Water is simply moving down its concentration gradient. Osmotic pressure is the pressure that develops due to osmosis. The greater the possible osmotic pressure, the more likely that the water diffuses in that direction. So let's take a look at this experiment. This diagram is actually very good. So we have a solution with some solute in it, right? So we have a 10% solution by weight. So we have a certain amount of sucrose, say, in here. We put it in another solution that's a 5% solution. Again, it could be something like sucrose. Sucrose's molecules are shown in green, the water in blue. So we can see there's clearly a higher solute concentration inside this flask. We have a membrane at the very bottom of this flask. It's a plastic membrane that's porous. It allows just the water molecules to pass through, which are small. It does not allow for the sucrose molecules to pass through. So water will go down. We'll move down from an area of high water concentration to an area of low water concentration. Now, eventually what happens is you can see what's happening is as the water moves, is that this column moves up, right? You can see that. And what we can do is actually measure the height of that column. That column has a certain weight, right? And because of that weight, it presses down, it causes pressure. And that is actually the osmotic pressure. So osmotic pressure is a real thing that's measurable, very easily measurable in a lab. So you can apply a pressure, and eventually this will stop because the osmotic pressure pushing down will cause a kind of hydrostatic pressure, in which the water molecules will actually move back across this semi-permeable membrane in the other direction, just due to the water pressure. And that will balance out the movement of water down its concentration rate. So what you would see in an experiment, take about half an hour, you'd see the water sort of magically moving up and up and up, and then finally it'll just stop and sit there forever, okay? So I like that diagram, it's very good, very good way of showing this. Okay, so we're going to talk about tonicity, which is another topic you may have heard of. You've probably all done in high school, right? Tonicity. Okay, so let's review it now. So tonicity is it's related. It's relative. It's the relative. Okay, it's the relative concentration of a solute outside this cell as compared to inside. So basically, it's a way of comparing solutions, right? That's how I would put it. It's a way of comparing solutions. So we're comparing a particular solution, typically with a cell, okay? So in an isotonic solution, and you can sort of picture in your mind an experiment. You're taking a red blood cell. We always use red blood cells. And you put it in, and you're a scientist or a doctor, and you put it in a small dish with some water. Now, if that solution in the dish is isotonic, and you wait 30 seconds or a minute or 2 minutes, you'll see that the cell doesn't change in size. Okay, there's no change in cell volume. Okay. Now that fact that the cell didn't change in volume indicates that there is no net gain or loss of water from the cell, and it also tells you something about the solute concentration of the cell relative to the solute concentration of the solution. So the solution and the cell have essentially the same solute concentration. Okay. That's what isotonic means. They have the same solute concentration. Now, again, there's a quick caveat I've got to throw in here, which is this only works if we're talking about solutes that can't pass through the membrane of the cell, okay? If we talk about solutes that can penetrate the membrane of the cell, this rule doesn't work. But for our purposes and generally when we're talking about these things, the solutes we're talking about don't pass through the cell, okay? So that's a little bit of a warning there, okay? But given that these solutes cannot pass through the cell and water is the only thing that can pass through the cell membrane, then this is absolutely correct. All right, let's go to the next one. So we have a hypotonic solution. You take your cell, red blood cell, you put it in the beaker of water, you're looking at it through a microscope, and you see that the cell actually gains water and swells up, and eventually may even lyse, which means that it breaks apart, okay? Lysis is to break apart. This occurs when the solution has a lower solute concentration than inside the cell. Let's draw a picture of this to see if we can understand what's going on. Okay, so we're going to have a hypotonic. So we have this. I'll draw water in blue. And we put our red blood cell in here. Red blood cells are biconcave. I don't know if you know that. They look sort of like that from the side because they're sort of concave on both sides. So we put our red blood cell in there. So we're doing the hypotonic solution. So our solution is hypotonic relative to our cell. What does this mean? Well, it means that over time, the red blood cell will get bigger and bigger and bigger, and eventually it might even pop, right? Now, why does that happen? Well, let's go back to looking at what's inside these cells. So essentially, a hypotonic solution is going to have fewer solutes than inside the cell. I'm going to use a different color here. I'm running out of color. I'll use black. So this hypotonic solution has very few solutes in it. It might even be pure water, right? So it would have none. And then the cell has a ton of solutes, which are indicated by these little black dots, right? So those are the solutes. Those are the dissolved substances. So tell me about the water concentration outside of the cell compared to inside. Where is the water concentration higher? Is it higher outside of the cell or inside of the cell? So the cell is packed with solutes. It's a red blood cell is probably hemoglobin, right? Protein for carrying oxygen. So the water concentration is much higher outside of the cell. Now, the plasma membrane of the red blood cell has aquaporin channels. Water can pass through the aquaporin channels. So water will move down from an area of high water concentration, right? See, I've drawn the solutes. That might be throwing people off here, right? The water concentration is high outside because there's very few solutes. The water concentration is low inside because it's just packed with solutes, like hemoglobin, things like that, right? So water, blue, so there'll be a net movement of water, it's supposed to be an arrow, into the cell, right? And it doesn't take very long. Within 30 seconds or a minute or 2 minutes, the cell is getting puffier and puffier and puffier. And eventually it may lyse, so it'll just completely break apart, right? Okay, potentially. Okay. Does that make a little bit of sense? So that's a hypotonic solution. Okay. So let's go back to isotonic a second, because we didn't really address that one. Isotonic would mean that the solute concentration is the same inside and outside of the cell, right? Isotonic. So here's our red blood cell. I'll make it big this time so it's easier to see. I'm using black for our solutes. Well, what I'm trying to indicate here is there's no difference. I'm just sort of making random dots, right? There's just as many per unit volume here. There's the same number of solutes inside as outside. That means the water concentration is the same inside and outside of the cell, right? So what is water going to do? Well, it can pass through aquaporin channels into the cell, but it can also pass out of aquaporin channels randomly out of the cell, right? So water's entering, water's leaving, but there's no difference, right? There's the same amount, right? So there's no net movement of water. Therefore, the cell doesn't puff up like the other solution. Let's do hypertonic. Okay, so this is the last one. I'll make my cells bigger. So I'll make my red blood cell bigger so we can see things better. Here's my red blood cell viewed from the side. That's why it looks like this funny shape. Okay. Solution has a higher solute concentration than inside of the cell. Okay, so let's draw that. So what the idea, I'm going to run out of dots here, right? But so I'm just going to do this around this hypertonic. I'm not going to do the whole thing. I'm going to be here all day, right? But I'm just going to do it right around the cell, right? We're indicating here that we have a lot more solutes per unit volume in the fluid that's outside of the cell compared to inside of the cell. So let's figure out what should happen here. So where is the water concentration higher? Is it going to be concentration higher inside the cell or outside of the cell? It's going to be higher inside, right? Because the solutes, there's not as many solutes here, so there's more water. Here, there's a ton of solutes outside. Those take up space, there's less water. So water, of course, is going to be moving into the cell. But overall, there's more water moving out because water's good. Because of course, it's a random process, right? Some will enter, some will leave. But overall, more are leaving, right? And therefore, what's going to happen to our cell? If we're looking through the microscope, we'll see that the cell loses water and it's going to get smaller and smaller, right? It will shrink and shrink and shrink. Right, and that's actually referred to as crenation. For our cells, so the red blood cells, it'll look very spiky. That's what is referred to as crenulated. Plasmolysis is something that occurs in plant cells. Plant cells have a cellulose wall. So with plasmolysis, what happens is the cell membrane can shrink and it'll actually shrink away from the cell wall, which doesn't shrink because it's very rigid. Okay, so that's what plasmolysis is. Okay, so basically, there's a video here. We've gone through. You can take a look at this video later, right? I've uploaded this for you. So here's our animal cell. Isotonic solution, water enters, water leaves, but there's no change in cell volume. Hypotonic solution, water enters the cell by moving down its concentration gradient because the solution is hypotonic relative to the cell. It has a lower solute concentration. The cell may get really puffed up. This is a red blood cell, puffed up in maybe lice. Here's the crenulated cell. You put the red blood cell in a hypertonic solution. Water moves down its concentration from higher concentration inside the cell to outside of the cell, and the cell shrinks and it gets crenulated. So it gets sort of spiky looking. Very similar in plant cells. Here it is. Your plant is happy. It's isotonic. Hypotonic solution, the cell will actually swell up. What's this? Oh, it's actually labeled here. This central vacuole, remember that holds water within plant cells. And then finally over here, we have plasmolysis where you put the plant cell in a hypertonic solution, like for example, a really strong salt solution. The central vacuole loses its water. The cytoplasm shrinks. This is called plasmolysis. So it's shrunk away from the cell walls. Okay. All right, so let's move on to another topic, which is transport by carrier proteins. So carrier proteins are required for what's called facilitated transport. Okay. So let's break this down. So we're going to be transporting things across the membrane. Okay, that's what the word transport word means. Using a transport protein. So facilitate is to make something easier. Does that ring, like, when you facilitate something, like, if you have a meeting, a person who's sort of leading them, like, not necessarily the leader, but they're helping along. So, okay, now this person's gonna speak, now next this person's gonna speak, they're organized, that's the facilitator, right? So facilitate is to make something easier, okay? So let's look at this. So we have facilitated transport, explains the passage of charged molecules, such as glucose or amino acids down the concentration gradient. So let's use the example of glucose. We talked about this before. We have glucose, it's typically in a higher concentration outside of the cell than inside. We talked about that, right? Because our cells use up glucose to make ATP. So glucose, in a sense, again, in quotes, wants to move down its concentration gradient from outside to inside, but it's prevented from doing so because it's a polar molecule. It's a relatively large polar molecule, a 6-carbon sugar with a bunch of hydroxyl groups, the OH groups, makes it polar. So we talked about this earlier. We have the glucose transporter, right? It's highly specific carrier protein that undergoes a shape change. We drew that on the board. Now, this process does not require ATP to move the molecule. Remember, I showed you how when that glucose bound to the transport or carrier protein, it then had a shape change, right? And that didn't require any energy. Okay, so that's facilitated transport. It's a form of what's called passive transport, as opposed to active transport. It does not require energy in the form of ATP or any other type of energy, right? Other than just the random thermal energy of molecules bouncing around, right, within your cells, which comes from the fact that you have a certain temperature within your body. Now, let's contrast to another kind of transport. You have active transport. This is where you transport something like a charged molecule or a large molecule like across the membrane. And we can actually move it from an area of low concentration to an area of high concentration. So this is essentially like if diffusion allows things to go in a sense downhill, right? This is like moving something uphill. It's going to take energy. So we can accumulate a molecule either inside or outside the cell. This is highly specific. It's going to require, again, a type of carrier protein, and energy is going to be required, often in the form of ATP, adenosine triphosphate. So let's take a look at this. So here's facilitated transport. We have here in this picture, we have a carrier protein. They've drawn it looking like a channel. It's not a channel. It's not open from outside to inside. So the artist did a little bit of a bad job here. Here's the solid molecule. It's binding to a specific region of the carrier protein. It then changes shape, and then the molecule is released on the other side. And then it goes back to this neutral position. Again, remember, this is not a channel. It's not open from the inside to the outside. So this process is passive. It does not require energy. The molecule is simply moving down its concentration gradient. So it's essentially just diffusion. All right. Then we have active transport. Let's see if we have a picture here. So active transport looks like this. Well, maybe we'll do the video so we can see.
When substances need to be driven against their concentration gradient from low to high concentrations, active transport is required. This process uses energy to move a substance across the membrane using selective protein carriers. The sodium-potassium pump is a specialized membrane protein carrier common in most animal cells that moves sodium and potassium ions across the plasma membrane. ATP fuels the pump in the movement of these ions from low to high concentrations, moving sodium ions outside of the cell where they become concentrated and bringing potassium ions into the cell where their concentration is higher relative to the outside. In other instances of active transport, the concentration gradient created from the action of one pump can be used as the input of energy to drive a different pump, a process known as coupled transport. The large concentration gradient of sodium ions is used to bring glucose into the cell against its concentration gradient.
Okay, so that was a really good example they happened to use there. So the first thing they talked about was a very famous pump that you have in all cells of your body, and it's necessary for life, which is the sodium potassium ATPase. Have you heard about this before? No, yes. No one's heard about it before? You didn't heard, but you, okay, all right, that's fine. Now you're hearing about it. So the sodium potassium pump is necessary for life. Every single cell in your body has one. Let's take your brain as an example. When your brain uses a tremendous amount of energy relative to other organs in your body, and about 70% of the energy their brain uses is just to fuel this one pump. Okay, it's so important. So why is this? Well, if you look at your cells in your body, here's a cell, it could be a neuron in your brain. There's an unequal distribution of sodium and potassium across the membrane. So the mnemonic to help us remember is pumpkin. Okay, so this pump, it pumps K in, okay? So K is potassium ions. So it's going to pump potassium into our cells. So we make, and I'm going to, concentration is often shown in the square brackets. I'm going to not do that. They're just going to keep it simple. So we have a large concentration of potassium inside of our cells and a smaller relative concentration of potassium outside. And at the same time, this pump actually takes sodium and pumps it the other way. So I'm going to draw a really large sodium outside and a small sodium inside to indicate that the concentration of sodium is much higher outside than inside. So this is kind of the opposite of diffusion. Remember, by diffusion, what should happen? Well, potassium should move down its concentration gradient out of the cell, and sodium should move down its concentration into the cell. And that, in fact, happens. There are sodium-potassium channels that allow for the diffusion of those ions. But here, what we have is a pump. So this pump sits in the membrane, and so sometimes you'll just draw like this, it's sort of symbolic, like an abstract way. So which way is it pumping things? It's going to move potassium into the cell, and at the same time, it's going to move sodium out of the cell. Okay, so this pump is active in all your cells. It's very tirelessly working all the time, and you can't just pump things without any energy. So what you're doing, you're having the hydrolysis of this very important molecule, which we talked about a lot, right? Adenosine triphosphate is hydrolyzed into ADP and inorganic phosphate, right? So that energy in that high energy molecule is being used up to do this, to move this pump. And that's what's being shown here. And what they're showing you in this complicated diagram, essentially is cycling. So the carrier has a shape that allows it to take up 3 sodium. Those 3 sodium enter into specific spots within this carrier molecule. ATP is split, meaning it's hydrolyzed, and you have then ADP and inorganic phosphate attaches to the carrier. Phosphate group attaches to the carrier. There's a change in shape now of the carrier molecule, similar to what we talked about earlier with glucose today. Results in the carrier releasing the 3 sodium now to the outside, right? So it's almost by magic. The sodium was inside, now it's outside, right? So we're moving sodium against its concentration gradient. You can think of this as like moving a ball uphill, right? You're moving something, taking energy to move something uphill. The carrier has a shape that allows it to take up 2 potassium from the outside. It picks up 2 potassium on the outside. It then the phosphate group is released from the carrier. The change in shape occurs, and the 2 phosphate ions are now released inside of the cell. Okay, so it's constantly cycling around and around and around, moving these ions across, maintaining this ion gradient. Again, potassium is very high inside, very low outside. Sodium is high outside and low inside. So if you remember, so again, just remember the word pumpkin. It's pumping potassium in, therefore potassium is higher inside than outside. Okay, we'll end there because we're over time. I'll see a good number of you today in lab. Remember that your pre-lab is due, right? As well as your post-lab for next week, right? Last week. Last week's post-lab is due and today's pre-lab is due. Today's pre-lab, as I mentioned in my email, You don't have to do this type, you can just print it out and then draw on it, right? Because you're labeling, it's a labeling exercise mostly. Okay, I see you guys in the lab and see the rest of you in the class. Next time.